sort of what the structure is, and I'll introduce our speakers. So Andy Kliskey, the director of Idaho EPSCoR, is going to sort of talk a little bit about what that project is. It might be unfamiliar to many of you here. Some of you might be familiar with it. And we're going to go through and do short presentations from sort of three different perspectives. One about social ecological systems science, one about trout genetics and habitat, and one about sagebrush genetics and habitat. And then at the end, we'll have a panel discussion where you are welcome to ask questions and make comments about any of these subjects or other things related to this research. Okay, let's see here. So we'll start with Andy Kliskey, professor in the Center for Resilient Communities at the University of Idaho and the director of Idaho EPSCoR.
Tēnā koutou, tēnā koutou, tēnā koutou katoa. Good evening and welcome. I'll give you a few words of introduction in New Zealand Māori. I'm originally from New Zealand, Aotearoa. I now find myself in Idaho and loving it. This is my home now. As David said, I'm both the Centre Director for the Centre for Resilient Communities, and for the last few months I've become the statewide director for Idaho's National Science Foundation funded EPSCoR program. I'm going to give you a little bit of an overview of what that program is, what it means for Idaho and Idahoans, and what we're doing in this current phase or funded program for EPSCoR. So EPSCoR stands for the Established Program to Stimulate Competitive Research, and it's a federally funded program to increase the research competitiveness of Idaho's universities, research universities and institutions nationally, to kind of bring us up to the level of the New Yorks and the Californias and the Massachusetts. And so there are a number of states, relatively developing states, many of the western states that are part of this EPSCoR program. And it does this by supporting research infrastructure improvement programs, of which we are one. And that means bringing in new ideas, new people. A big part of it is the human capital that it brings into Idaho, into its universities. And some of the folk here today, Sarah Ebel is part of the new influx of ideas and brains to Idaho. Some of it is about facilities development and some of it equipment, but it does it through a cohesive research program. And the way that happens is through funded research programs, typically what we call a Track 1 Research Infrastructure Improvement Grant. And the current one that we have is called GEM3. And this, we're just in starting year 2. It was funded in October last year. And what ends up being in a 5-year research program is decided in part by the ideas that faculty across the 3 institutions bring together, but there's also significant input that comes from the state of Idaho through the State Board of Education and through a statewide Idaho committee. And their charge is to provide input to the program on what are some of the key research needs, some of the areas that we need in research competitiveness improved in, and areas that can enhance workforce development in the state of Idaho. So an important part of it is making the link between research and the universities, education, career development, and workforce development. How will it enhance job creation for Idaho in the long term? So that's kind of the really big picture about EPSCoR. And as I said, it does it through a number of mechanisms, but the one we're going to talk about today is a 5-year program of which we're just entering year 2 to really develop and enhance some science, so education, job development, partnerships across the state, not just within the universities, and it's really a really phenomenal process of collaboration. So this current GEM3 is part of an award of $20 million from the National Science Foundation with a match of $4 million from the state of Idaho through the Higher Education Research Council. So $24 million, of which $4 million comes from all of our state taxes. So it's a partnership between state and federal. which is important to understand. And the broad topic that our current one is this GEM3, G-E-M. I pronounce my E's like you hear I's and vice versa, so it sounds like I'm saying the boy's name Jim, but I'm meaning GEM as in the GEM state. Genes to Environment, Modelling, Mechanisms and Mapping. That's actually our short title. We have a long title which I can't remember and we're not going to go into today. If someone wants to know, I'll look it up. And the broad theme is about advancing fundamental knowledge to predict how plant and animal organisms adapt to external stresses and a changing environment. And we'll unpack that a little bit, what we mean, but there are these three connected pieces. Identifying genome to phenome mechanisms for some plant and animals. Genome is the genetic makeup of an organism. So it's about the genetic makeup. Phenome is the physical and ecological and physiological characteristics of an organism in its local landscape. And so we're looking at that marrying up between genetic makeup, what's underlying our genetic makeup, or the plant and animal's genetic makeup, and its characteristics in the landscape. And modelling future adaptive capacity of the populations of those organisms, and I'll get to what those are, trout and sagebrush, as we've already heard, and then mapping those in the landscape and engaging with stakeholders, community members, state, federal, government, non-governmental organisations to better understand what that means in the landscape and for enhancing management decisions in Idaho's landscapes. It's a very interdisciplinary, integrative project, so there's a number of facets of science that are involved: genomics, ecology, landscape, social science, stakeholder engagement, plants, animals, and so a great high degree of integration that's necessary and convergence of these multiple strands of science. We're looking at two focal species. We can't look at everything, and so we've narrowed down to two focal species, one animal, one plant. The animal species is an aquatic focal species, red band trout, the terrestrial sagebrush, and they're both species that are important to Idaho. They're important for cultural reasons, for economic reasons, and so we've chosen those two. There's also an existing wealth of expertise among researchers in state and federal organisations, in our universities, and so we can kind of leverage existing knowledge as part of this effort. And then look at how that links to decisions and outcomes in Idaho's landscapes. Some of the detailed science that we're doing is broken down by three components. The modelling, mechanisms and mapping, that's the 3 M's that we refer to. And understanding the mechanisms, and so that is the genetic to physical ecological mechanisms, or the genome to phenome mechanisms in red band trout and in sagebrush ecosystems and sagebrush plant populations, require this in-depth laboratory and field experiments, sometimes referred to as common garden experiments. You'll hear referred to in both Shawn and Jen, who are going to talk about trout and sagebrush, are going to give us a little bit more information and detail about what that means. There's some computational modelling. Those models are population models. Where do particular species for trout and for sagebrush exist in the landscape currently? Oops. I lost it.
We'll get it back. Keep talking.
I'll keep talking.
There you go.
So using these computational models to look at existing distribution, and then also doing some near future projections into the future, maybe 20 years, to look at how those are likely to change over time given our current knowledge of changing landscapes, changing climate. And then the third piece is the mapping piece, and that is as it says, or you'd think by the term geospatial mapping, looking at what happens in the landscape. But an important part is also mapping it to different community groups, different stakeholder groups. And so the mapping piece here involves a very distinct and explicit process of engaging with decision makers, land managers, landowners, ranchers, other community representatives and groups across some of Idaho's landscapes. So we're going to hear a little bit about how each of these three pieces are starting to be understood and developed and studied for red-band trout on the one hand and sagebrush on the other. That necessarily means there's also an attempt that we need to do to integrate the understanding of both of those for broader understanding. So trout, sagebrush are kind of these model organisms that we're looking at, and there will be parallels to other organisms in the landscape as well. Okay, all of this work is engaging teams at Idaho State University in Pocatello, Boise State University in Boise, and at University of Idaho, Moscow, and some of its outlying centres. And so it truly is a collaborative effort across the major research institutions of Idaho. Also, some of it's primarily undergraduate institutions or colleges. College of Southern Idaho, College of Idaho, College of Western Idaho are involved, and then a number of state and federal agencies and other partners. And so just sort of in summary, before we delve into the first of the next three talks, Idaho's NSF EPSCoR program and this GEM3 is an effort in trying to build research competitiveness, enhance our knowledge, enhance the ability for the state of Idaho to undertake improvement in our understanding and to enhance workforce development through a collaborative program that integrates multiple strands of science and knowledge and converges those together to enhance management decisions and make a difference to Idahoans and to Idaho's landscapes. So that's kind of the overview of the program, and we're now going to move on to some trout work. David.
Go ahead. Your name's right here. Shawn, you want to introduce yourself?
Absolutely. Good evening. I'm Shawn Narum. I'm with, as it says here, the Columbia River Intertribal Fish Commission and adjunct faculty member at the University of Idaho. And so I'm your designated fish guy tonight, so I get to tell you some fish stories, and they told me to keep it short because they want to let some other people talk about plants and some other things. But I have the opportunity to start off by showing you some really great pictures of some of our native salmon and trout that we find in the Columbia River Basin. So what I'm showing you here are a map of the Columbia River Basin. I think most of us know that waters in Idaho drain into the Columbia River and out into the Pacific Ocean. And here I'm showing some of the native salmon and trout. On the left are the native anadromous salmon. So anadromous means essentially it's this life history of these fish. So they're born in freshwater. After about a few months to a year, they migrate out to the ocean. They spend a couple years in the ocean, then they return as adults to spawn back in freshwater. So that's that anadromous description. And so those fish are on the left there. I'm sure some of you in the room recognize them. You don't have to go too far from Ketchum to find some of these salmon. Just over at Galena Summit near Stanley, some of you may go there and actually have the opportunities to fish for Chinook salmon and steelhead. So those are returning up to the upper salmon area there, including to Sawtooth Hatchery there. Of course, the third one on the list on the left there is sockeye salmon. They're spawning and returning to Redfish Lake, another area near Stanley. And then you have to go a little farther to find coho, northern Idaho into the Clearwater drainage where you'll find coho salmon in the state of Idaho. Over on the right, I've got a list of some of the resident trout. By resident here, I'm just meaning those that spend their entire life in freshwater rather than moving to the ocean. And so you'll probably, again, recognize some of these species on the top right. Of course, some of these species are here in the Big Wood River, the rainbow trout and the inland red band. I do want to point out, and I'll touch a little bit more on this, is that those coastal rainbows that we find in the Big Wood aren't native to this area. It's actually the inland red band that are native. So the coastal rainbows in the drainage are found in their native areas much farther near the ocean on the west coast there, and I'll touch on that again in a moment. Continuing down the list, some other species, again, that are pretty common in this area. Cutthroat trout, very widespread and abundant throughout the state of Idaho. And then the last one on the list is bull trout. Bull trout are these more rare species, but they're found in these very cold headwater environments, including some of these high mountain streams in this nearby area. This GEM3 project is focusing largely on these inland red band trout. And I wanted to draw a quick distinction between these just to help people understand the difference between them. They are both the species Oncorhynchus micus, so I've listed that there at the top for those of you that get into Latin names, probably not too many folks. But what we have on the left are what's known as coastal rainbow trout or steelhead. And so these are actually a different subspecies than the inland red band trout that are on the right. And if you can see the different colors on the map there, those in pink are the native distribution of rainbow trout and coastal rainbow trout. So they're all along the west coast there. And it's in that orange area where we see all the native inland red band trout. And so it's a kind of a subtle distinction. A lot of people actually refer to red band trout as rainbow trout because they are the same species. They're a distinct subspecies. And so I wanted to make it clear that when we're talking about red band trout, we're talking about this very similar but distinct subspecies of rainbow trout. And the reason this GEM3 project is really focused on red band trout is not only because they're native in this area, but they occupy these really fascinating distinct environments throughout the state. So as we look at this map of southwest Idaho, we see that these red band trout are found in both desert and montane streams. We have, of course, the Wood Rivers represented on there somewhere. Let's see if I can find the laser. So the Wood River is here, and of course that's one of our montane streams that's represented where red band trout are occupied. But also of interest are these desert streams in the southern part of the state. So the Bruno drainage, the Owyhee Basin, some of these places that you may have visited in the past, these very low precipitation, high desert streams. And these are particularly fascinating because these red band trout are occupying these streams where water temperatures in the summer can reach up to 85 degrees Celsius, or Fahrenheit in this case. And that's important because in the species in general, these fish become stressed as water temperatures are approaching and getting above 70 degrees Fahrenheit. And so trying to understand how these fish have been able to adapt and to tolerate these really warm desert streams is something that's of great interest, especially as we consider things like predictions under scenarios of climate change suggest water temperatures are going to increase. So we want to get a better understanding of how these red band trout have done this. So this gets at one of the central questions of this GEM3 project for the trout component anyways. And what I'm showing here is this idea that there's some potential for thermal adaptation within the species. And so this graph is just trying to indicate that there's a range of thermal tolerance for the species. There may be some average upper thermal tolerance of about 75 degrees for the species. But within this species, there is some variation where in some of these desert streams, there's the potential for them to adapt to have a higher thermal tolerance on average than the typical montane stream. So this is actually a picture of Little Jackson Creek, one of these desert streams in the Bruno drainage that we're studying. Likewise, it's also worth noting that there are some very cold headwater streams where these red band trout also occupy. And so they may have been forced to kind of become specialized to perform well in those really cold environments. And so in those cases, we might see that thermal tolerance has actually been selected and shifted towards to the left in this case. And like I said, so this is kind of at the center of a lot of the trout work, is understanding this potential for adaptation in red band trout. And so this GEM3 team is actually capitalizing on expertise throughout the state. You heard Andy talk about the different researchers at the different institutions. Here I'm highlighting some work that on the mapping component that's being led by Donna Delpart. So she's with Idaho State University. She was scheduled to be here today actually, but she's out doing some of this work right now today, and so she wasn't able to be here, but she shared these slides with me. So what her specialty is in geosciences, and this is a aerial drone image. So she's flying these aerial drones to help us better map some of these areas where we maybe have limited access points in a lot of cases, so they're in very remote areas. And so in this case, what this picture is showing is these canyon rim walls here. So these are these desert canyon cliffs. And then we're able to quantify and map the habitat along the stream bottom. You can't quite see the stream bottom here, but on this next slide, what Don has been able to do with some of these drones is include a LiDAR camera. So this is a very specialized camera that can detect infrared, and so they can measure water temperature using that infrared sensor. And so I'm going to zoom in a little bit farther on one section of this, and hopefully you can see the different colors here. And so this follows the stream profile here. So this is Big Jacks Creek, and this thermal layer is representing water temperatures. So this was a flight that she did with this aerial drone survey at the beginning of summer. And what we start to see is water temperatures are ranging from the scale here in blue from the mid-60s, and some areas are fairly warm to the mid-70s. One of the things this helps us identify is that indeed there are some cases where there's some really warm water where these fish may be becoming stressed, but you'll notice this little blue pocket here may be a thermal refuge for some of these fish. So they may be able to move into these really cooler waters and be able to get a little bit of break from that stressful water temperatures that they're occupying. A lot of this in some places like this may be due to upwelling of ground spring water that's kind of flowing into the stream and provides these little pockets of refugees. So this ability to fly with an aerial drone is a much better way to kind of map this than some of us that are wading through the field in our waders and dipping a thermometer every few meters in the water. The next piece I wanted to move on to is trying to understand. So like I mentioned, there are the cases where fish can actually move and find colder water, but we do know that from a lot of our fish surveys in these desert streams, they're forced to deal with warm water. And so what I'm showing here is what happens to these fish and how do they deal with these warm water temperatures. One thing that we know, a fact and property of water, is that as water temperatures increase, the amount of oxygen available in that water decreases. So that's just a natural function of the water. So fish, in particular trout, they're highly mobile fish. They require a lot of energy and a lot of oxygen. So that's a problem for fish. So what they do, of course, I just back up for a moment and mention that, of course, fish don't have lungs to take up oxygen from the water, right? They have gills. So their gills, it's a layer here on their side of their head here, and there's a gill cover, and they're able to move that back and forth. That forces water across their gills. Those gills allow them to extract that oxygen from the water. And what I'm trying to show here is this kind of interface where the water would meet the gills right here, and the oxygen would go into the circulatory system. And the other thing I wanted to point out here is not only is it the oxygen now in the circulatory system going to the tissues and the organs, the primary mechanism for delivering that oxygen is the heart. So as fish experience low oxygen, what they do is actually increase that heart rate to deliver more oxygen to the tissues. And so this is actually one of the mechanisms of the GEM3 project that we're trying to understand more carefully, is understanding the performance of the heart when these warm water temperatures are experienced. Here I'm emphasizing some work from one of my close colleagues, Dr. Zhongqi Chen. He's a postdoc at University of Idaho. And what Zhongqi has done over the years when I've been working with him is developed this really fantastic electrocardiogram, or ECG system, to measure the heart rate of these fish. So those of you that have I've seen some interesting things going in the audience of recognizing that ECG is some things that some of you may have been hooked up to in the hospital to measure your heart rate, or some of your children's, or other people that you know. You may have just even seen it on TV, but the idea is you can measure the individual heartbeat of these different fish. And what we do is essentially we anesthetize or we sedate these fish in water so we can have these probes on them, and we increase the water temperatures, and we can measure the actual heart rate increase over time by the fish and understand how different fish are able to do this from desert or montane streams. And we do this in what's known as a common garden environment. It's something Andy pointed out earlier. And so this is one mechanism. Another mechanism that we're trying to get a good handle on is what are the underlying genes responsible for some of these traits that allow these fish to tolerate warmer temperatures. What I'm showing here is, so this is a particular plot that shows all the different genes across the chromosomes in the DNA of this organism, so in red band trout. And so what we're generally looking at is low levels of significance across the chromosome, but what we do have is this one peak in blue, and this is a strong candidate gene, it's known, that's maybe related to this trait of heart performance. And so as we start to understand some of these genes and how they might be useful for these fish to adapt, what we want to do is utilize that information for some of our long-term objectives. And those long-term objectives I'm just listing here as one of my last slides. And this is where we can develop some of these tools to screen natural populations of red band trout throughout their range. And the idea is to be able to use some of these genetic markers to get a good understanding of the potential for these fish to adapt to different environments. And so this is where the third M, or the modeling component, comes in, the statistical models that kind of take all this data, whether it be genetic, physiological, or mapping data, and help us understand which populations may have the potential to adapt under scenarios of climate change. And so you can imagine, if we have all this modeling data under scenarios of climate change, there may be some predictions that say some fish are likely to persist under scenarios of climate change given their existing genetic makeup, and there may be some populations that get wiped out if the water temperatures get too warm. And so the idea is to work closely with some of our stakeholders throughout the state. So that includes Native American tribes, a lot of people that I work with closely. It includes Idaho Fish and Game, a fisheries manager within the state, and some of the NGOs like Trout Unlimited and others that are working to, kind of conserve some of these fish and help them inform some of the management and conservation actions that they might want to take. So that's my last slide. I'm going to ask the whole questions. We're going to have a panel at the end, and so we'll take all questions at that time, but I'll pass it on to the next presenter.
Dr. Jennifer Forbey from Boise State is going to talk about sagebrush.
All right.
And she's got
I've got activities. Yes, I've got activities. So my name is Jen Forbey. I'm a professor at Boise State University, and I'm going to start with a bit of a confession of, so I did my graduate work at University of Utah. That's not the confession. The confession is that I would come to Idaho all the time to run the Middle Fork, the Selway, the South Fork of the Boise, the Payettes, and I would drive across and I would look out at the Sagebrush Sea, and I would go, ooh, as I was heading to these mountains. And so that's my confession. But then after coming here, I actually work on the animals that eat toxic plants, sagebrush being one of these things. And so as I came here, I see sagebrush as the sort of girl or boy in your classroom that, when you're growing up, before you sort of found your mate and you sort of ignored them, maybe they were a little plain. But then as you got to know them and you sort of went out a little bit, they had this personality. And then you finally started to realize that you're so interesting. And then you wanted to spend more and more time with this individual. And I feel that way about sagebrush. So I'm like obsessed with it. It is so fascinating. And so I hope today that I'm going to share a little bit of my obsession and why I think this species is amazing. And it's a story, a sage story, hopefully, of ancestry, which is the genome, the genetics of this plant, and maybe some things you don't know about where sagebrush came from. It's personality, as I call it, which is really chemistry, and that's the little activity that we're going to do, is getting you to know sagebrush's personality. And I've got a couple volunteers that I'll walk you through what we're going to do with this, but I'm going to help you smell, see, and taste the personality that is sagebrush. And then talk a little bit about how we might use these two things, the genome, the genetic, this ancestry, and the phenome, the sort of personality of a plant, to make, potentially improve the way in which we conserve the species. And so I'll start with the importance of ancestry. So this is something that maybe many of you have either tried or have thought about doing and maybe were too afraid to figure it out. But we're very interested in where we come from, right? And we should be equally interested to know the organisms that we're trying to manage, where they come from. And so we're interested in knowing where we come from because we're interested in the genes that might make us more susceptible to particular diseases, and then we can change the management of those diseases by behaviors and changes in activities. And it also might explain our personalities, right? It might explain some of our mental states. It might explain some of our physical states. And so these things become really important to interpreting how you interact with your environment and the people around you. And so we use this information to make decisions. It helps us predict our health. And the old way, maybe the current way of doing medicine, is that we have this diversity of individuals that all come with different genes and different personalities and different physiology, and we tend to have single therapies, and then we get effects that some respond positively, some have no effect, and some have adverse effects. And where personalized medicine is going is actually being more personalized diagnostics, where they take these diverse individuals, you identify your genes, you take biomarkers, usually these are chemical biomarkers in your blood or in your tissues or in your urine, and then they give you more personalized therapy that hopefully improves the outcomes. And so this is the sort of approach that we can take with both red band trout and sagebrush by understanding the genotypes and also these sort of physiological or behavioral biomarkers of the organisms to make more personalized management decisions. And so now I'll tell you a little bit about sagebrush's ancestry, because we need to know where it came from to understand what it's doing now. And so this is a long history to start with. And so if you may not know that sagebrush came across, so the old world from Asia is where most of the artemisias, or at least the ancestors of our sagebrush, came from. And it came over based on pollen records in 2 waves. The first wave was 28 million years ago. The second wave was 1.8 to 11,000 years during the Pleistocene. And they know some of this based on pollen records. This part's pretty fascinating. So they found mammoths and also prehistoric extinct horses. And in the stomachs of these horses, what they find is Artemisia frigida. So they were eating Artemisia. This is an herb, so it's actually a soft Artemisia type, so it's not, or woody type. And then they also found Artemisia flowers in the stomachs of these horses, and then in the same layer geologically as the woolly mammoth. And so they knew over this period of time that sagebrushes were over in North America coming from Asia. And what happened during that period of time is back and forth between the old world to the new world, and a lot of hybridizations. And what happens so our genomics, our plant genomics said that what happened in North America, which happens to many North Americans, is sagebrush got fat genetically. And so because of this hybridization, he's from Swiss, and he asked me if that was okay to say, and I said, I think I can say it, but you can't. So it carried around a lot of genomic baggage, and I think that's what made it, we think, adapted to a variety of climates. And during that period of time, there was a variety of climatic and environmental events, from really high temperatures to droughts to cold temperatures, and that created diversity in the genome of the species in North America, more so even than in Asia. And so with that diversity comes these unique personalities. And so I'll talk a little bit about how we might be interested in the ancestry to understand our genomes and make predictions about our chemistry or our biomarkers. And this is the genetics of sagebrush. And I know it's super tiny, but it's because it's very complex. So this is including all the old world and new world species. And I'll just point out some of these really unique and I think interesting personalities. So I'm going to highlight one that you may be familiar with. This is you all enjoy art. So Van Gogh was known to be addicted to absinthe, which comes from the absinthium clade, which is an old world version of Artemisia. So it's Artemisia absinthium. And it might have created unique ways of him looking at art. So that has a benefit of Artemisia. The second is the Eurasian clade. So this is Artemisia annua. And Dr. Yo-Yo was given the Nobel Prize for relying on Chinese medicine to discover Artemisia sen, which is a compound that's found in Artemisia annua, and it's the #1 anti-malarial drug that's still used today. And our species of sagebrush contains similar classes, similar to Artemisian here in North America. I keep using the wrong one. And then this is, oh goodness, this is a bit messy, but these are our group. So this is the New World endemic, so you might know some of these. Artemisia obuscula, tripartita, which is 3-tip. Here's our big sagebrushes, black sage, California sage. So this is our local populations that we might be familiar with. And they come with particular phenotypes that we try and use. So we know very little about identifying genetically who these organisms are. Instead, we rely on morphology, which can be extremely challenging to do. And part of the reason that we're interested in this is that personality, at least my interest, and some maybe of our sort of people that used to use these rangelands and maybe still do, is that different species and populations have different levels of palatability. And the wildlife and our domestic species know this. There are particular populations and areas that they'll eat it and others that they won't. And so this is the personality that is the chemistry of sagebrush that drives these foraging decisions and makes some plants palatable and others not. And wild things and domestic species aren't the only ones that can listen to these sort of chemical personalities. So the Native Americans actually have a long history of using sagebrushes. So this was a paper in the late 80s. And this is just one example that the Shoshone and the Paiutes were using sagebrush for a variety of medicinal purposes in native lands. So now we're going to do the activity to see if you can tell the difference. And so what I have is, and I'll walk around, and I'm going to put one group here. So we have black lights, and what the helpers are going to do is that you have two vials. This is chemistry 101 going on here. So you have two vials, one that says ARB for Artemisia albuscula, the big vials, and one that says Wyoming, and they already have water in them. It's just water, not ethanol or alcohol or anything else, so don't worry about that. And what you're going to do is inside, I brought some leaves. Find my little bag of leaves. So inside are smaller vials, and I brought gloves in case you want to be serious chemists. You can wear gloves. But so this is what you're going to do. You're going to dump the RB into the RB. Right, so you're going to dump a few leaves. Just a few of them, because then you're going to pass the vial around. And those of you that are brave enough, so if you're not very brave, you can just smell the difference between the two vials. You're going to make us these. If you want to actually see what a sage grouse or a sheep tastes, you can take a single leaf, that's all I recommend. A single leaf of the ARB, which is the Artemisia arbuscula, and a single leaf of the Wyoming. Keep them in two different hands. And taste each one of them. I recommend the Wyoming first, followed by the arbuscula, but you can choose any route that you'd like to take. So after our volunteers have, wait, I have one more. Oh yeah, you've got one. So after you've dumped the right name, a few of them in the vials, and we're going to let those simmer because we're going to see what a sage grouse looks like when they fly over a landscape and sees these two species. You can actually tell the difference between them. When you're done with that, pass the vials down to some folks. You can take out a leaf, dump it in your hand, taste it, smell it. You should be able to smell the difference between these two species. You will definitely be able to taste the difference between these two species. And after our vials are covered, you have a black light in your bag, and then you're going to be able to see the difference between these two. So the palatable species tend to have more coumarins, the type of chemical, that fluoresces under UV. And sage grouse and pygmy rabbits and sheep and mule deer and pronghorn all prefer the plants that have UV light, that they fluoresce. And we think that they're actually able to see these plants. I don't know what it's telling them, but it's telling them something. This is the personality that is sagebrush. So once you have, maybe what I'll do is I'll hold up, can I just see your eyes real quick? What you'll see, don't flash it in your eyes, please. That you'll see. Ooh. See this UV reflected? And my Wyoming is actually fluorescing a little bit because I had these sitting 2 hours ago. So over time it will absorb, but what you'll see is that the arbuscular one will fluoresce more. It's like a phosphorescent bay in an ocean than the Wyoming, so shut that off. So if you'd like to shine on yours and hold it up and see if we can see something. Oh, much better. See? Yeah, so that one fluoresces right away. And so if you guys can do that, go ahead and see if your black lights, some of them, they operate differently. Actually, and then some of you have a long UV light. So now that I've distracted all of you, so you can pass them around, make sure, so does anyone taste it? Anybody willing to taste the leaves? Okay, so as they come here, I have 2 more. I'll put them over.
There we go.
So take a leaf. There's receptors in the back that are the bitter receptors. That's where it'll really hit you. If you choose to keep it in the front of your mouth, you won't taste it quite as much. But definitely the arbuscula, the little tiny leaves, those are much better tasting. Almost, I don't know, I think it's a little minty. So now, hopefully you now know how animals and organisms might use this. As you're looking at it, I'll tell you one story very quickly, and that is about Sagebrush uses these chemicals
Tasting right now.
partly to tell other organisms not to come eat me. They're typically not telling you, come feed on me. That's not a great thing for a plant. But they're using them to communicate with each other. And so this becomes the communication and how sagebrush tells its other buddies that there are bad things coming. So they emit these volatiles, which is the part you smell, and they're telling their related plant species information. And what's important about this is that it has multiple personalities, so it can induce and change the chemistry. And plants that are younger and are more genetically related can hear the chemical message and respond better than plants that are not genetically related. So these plants are talking to one another. And the interesting part is that if we disrupt the genetics of these plants out in the field, we disrupt the communication that these plants have relied on since they came across Beringia, the Bering Strait, to talk to one another and tell them about their environment and actually be adaptive to what's going on. And these are some of the things that we've done over the recent to disrupt this genetics and personalities. So we used to chain sagebrush off the landscape. That's one thing we've done. We kind of continue to do this. So some of our agencies still mow sagebrush. So here you can see that occurring in Wyoming, and it results in this. So now you have fragmentation of communication of species. We can have natural disruption, so the fires that are coming can result in changes in our landscape. And what is important to note is that these are rapid disruptions in both the genes and the personalities of these plants. So what habitat could look like this today, and when a fire comes through, it looks like this. And then over time, the plants come back. So this is a plant that survived the fire, and here are all these little cute baby sagebrushes that are coming back that probably belong to this plant. But when we go to restore, we're putting out seed from places that we may not know where it comes from. And so you might have remnant populations that have these genotypes that have maybe evolved or been in these landscapes for long periods of time that know how to talk to one another because they're the same genotypes. And then when we come in and reseed, these are sort of representing different genotypes. We might be putting mixtures of sagebrush together that don't know how to talk to one another and therefore are not as well adapted to the environment. And so what our group would like to do is, can we predict these genotypes? And I'll just briefly show you that we use common gardens. We, I don't know if I can actually use that. I won't use that, but maybe there we go. I'm going to show you what we're doing with our flights. There it is. So this is some footage of our aerial drones, unmanned aerial systems, where you can see a garden of sagebrush. These are a variety of different genotypes, so different genes thrown in together and saying, how do you talk to each other and what's going on? And so we're flying over, we're actually listening to the sagebrush right now. We're collecting some samples to measure its chemistry and see what it's saying. And you can see an extant population of sagebrush that's native here. We then can take that information and we can create 3D models, so structure of the plant, so we can get volume and biomass of these plants, so we can predict the growth rates of these plants. We then can take hyperspectral, so it's the wavelengths of light that gives off the chemistry of the plants, and we can use that to predict genotypes of the plants through these hyperspectral images that get out. We can measure the stress of the plant through thermal imagery, so these are, the red ones are the stressed plants in a warm environment. We can also use this to measure propagation. So this is a remnant population of sagebrush that survived the fire in the soda fire complex. This has been reseeded, and now we can measure which plants come up, how fast they grow, whether they survive, and what they're saying to one another based on those chemical profiles. And our goal is to not do this one management fits all, because it doesn't, but to actually take this known genetic diversity, these different ancestries of our sagebrushes, understand the chemistry, their behavior, their physiology, and create the right combinations in the right environment so that we can improve our management tools and the outcomes of those management. And that's what our GEM3 group is hoping to do. We hope to have partners like you thinking about these things and helping us survey, and we look forward to questions at the end. So thank you for your attention.
Okay, hi, my name is Sarah Ebel. I'm an assistant professor at Idaho State, and I'm going to try to be thorough yet quick so that we have enough time to have all of your questions and attack that thoroughly. So I'm a member of the Socio-Ecological Systems Working Group as part of GEM3. I'm an anthropologist, so I study humans, and which is very different from trout and sagebrush, but the people that manage trout and sagebrush are humans. So adding humans into these systems complicates them even further than what Shawn and Jen just talked about. So we need to understand who's involved in management, who cares about these systems, why do they value them, and how do we move forward with new management plans, possibly new management plans, in light of a changing Idaho landscape. Oh, is it this one? Oops. Okay, so one of the overall objectives of our socio-ecological systems team is to understand the impacts of environmental and social change on Idaho's landscapes, wildlife, and people. So we're really interested in not only how trout and sage adapt to land use change and climate change, but also how are people influencing that adaptation, and how are those landscapes actually influencing people's decision making? Because although we might not notice, we are actually getting feedback from ecological systems all the time, and it's influencing our behavior. So we have a number of overarching research questions. You can read them there, but most of them are focused on trying to understand local knowledge of systems, so working with stakeholders to see what knowledge do they have of sagebrush or trout systems, how are environmental or social factors, including development, tourism, farming, affecting these species and those species' adaptive capacity, and then how do stakeholders value these different landscapes, and how are those values understood and then implemented in management, if at all. We're also interested in how those values influence decision-making in terms of both land and water, and what might be barriers and opportunities for decision-making or changing management plans. And lastly, we're interested in gathering all of this social data within these systems and integrating it into these ecological data in order to create scenarios, which are really trying to understand in future climate forecast models, what are landscapes in Idaho, what might they look like, and then what might that mean for humans and land use change and land use management of these systems. One way that we do this is really trying to engage very closely with stakeholders. So stakeholders is what it sounds like. You guys are all stakeholders. Anybody that has a stake or care in one of these problems. So we are holding, and just started last May, holding stakeholder advisory groups. And these are groups of individuals that are interested, again, in this problem. So either they're land use decision makers, managers, landowners, people from nonprofits and land trusts, individuals who might want to collect data on their land. And these people are coming together to help us really form questions. So we're trying to collaboratively form questions with stakeholders in order to inform our own research moving forward, so that this research is not only ecologically or scientifically appropriate, but also socially appropriate in trying to solve some of the issues that everyone sees happening in Idaho. One other way we are engaging stakeholders is through CBONs, or community-based observation networks, and these are very long-term collaborative groups that have been coming together they're collecting data, and you can correct me if I'm wrong, David, collect data on their own, coming together to talk about that data, share that data, and really creating a collaborative effort to basically have citizen science in many ways, where citizens are collecting data and addressing problems that they want to be addressing. So we have a number of different projects that are just starting, and we have two excellent students that have just started, one a master's student and one a PhD student. So I'm just gonna briefly talk about the work that they're doing. Lizzie Josie's a master's student of science at Idaho State, and she is interested in working in Teton Valley, and she's looking at the effects of land and water use management on populations of Yellowstone cutthroat trout. And Yellowstone cutthroat trout are a species that are threatened, I believe. Is that correct? This is not my expertise. In Teton Valley. So one of her overarching research questions is does changing stream connectivity affect these Yellowstone cutthroat trout populations? So stream connectivity means, are the streams flowing, and is it allowing populations to go up into montane streams? Again, this is not my expertise. And is it allowing the populations to go back down, or are the streams drying up in the summer and therefore separating these species from possibly where they might spawn or have some of their natural habitat? The other issue with this is that Yellowstone cutthroat trout are not only affected by possibly drying streams, but they're also affected by brook trout and rainbow trout and hybridization of these trout species. And so connectivity is a stakeholder-driven question from Teton Valley, and stakeholders are really interested in if we restore or construct connectivity of streams, can we actually bring back or improve Yellowstone cutthroat trout populations? So on the human side of this, Lizzie's looking at, are there decisions that individuals or groups are making right now, institutions are making right now, that are affecting these populations, and can those decisions change, and what should they change to if we want to actually restore some of these populations? So we have another student who's working with Kelly Hopping at Boise State as part of the sociological systems team, and her name's Haley Netherton. And she is really interested in values, so human values, conservation values, environmental values of individuals in Teton Valley and across Idaho, and of sagebrush. So we learned about sagebrush. Sagebrush is very important. There's many different types, but it's being threatened by farming, fragmentation, oil, natural gas, all of these different human-caused problems. So what she's really interested in are the local preferences for sagebrush? Are there local preferences across Idaho? Do people care about sagebrush? Do they value sagebrush differently across Idaho or even within communities? And what does that mean for management and adaptation of these systems long-term? If she's interested, is there potential conflict that might arise if there's one group that highly values sage because they love bird hunting, but there's another group that is pursuing some type of industry that needs that area and might destroy the sage, or questions like that might arise in stakeholder groups. And this question of sagebrush values was also stakeholder-driven. It came up in a stakeholder advisory group meeting in Driggs back in May, and this SAG, we call them, Stakeholder Advisory Group, was really interested in, if we want to conserve sagebrush, well, one question, do we want to conserve sagebrush? Two, if we want to conserve sagebrush, what happens if people value it differently? And so can we map people's values across the landscape? These are examples here from different places, I guess. Is that? Anyway, that looks like Alaska. Yes, sorry. I need my glasses. So she's really interested in how do we actually get at these values? How do you understand human values, and how do we map them across space? So very briefly, I'm going to talk about some work that I've done. It's not in Idaho, but one interesting thing about academic questions and the problems that the world is facing is that they're not overly unique when you get down to brass tacks. So people's values matter because it influences their behavior and their motivations. So when you're thinking about conservation or adaptation to climate change, people's objectives and what they want out of adaptation are going to differ across geographic space, but they also differ across cultural groups. And even within Ketchum, Idaho, there's multiple cultural groups. And so those differences in values might actually cause conflict when we actually think about how we're designing management plans and how we're thinking about adaptation long term. So I'm just going to briefly talk to you about some work I've done in Chile. I've been working in Chile for 10 years, and there was a conflict that came up in one of the communities I work in, very small community, about Ketchum size actually, 3,000 people. And the indigenous community there wanted to design a protected area. They saw it as a form of adaptation to environmental change, where the non-indigenous small-scale fishers did not want a protected area because it was threatening what they see as threatening their livelihoods and their access to resources. Oops. So I asked how do underlying human values shape preferences for adaptive governance? And governance is not management, but governance is stakeholders coming together to attempt to work out a plan long-term for resources or any type of government action. So I work in Chile, again, southern Chile right here. This is the island of Chiloé. I'm not going to spend too much time on this. It's beautiful down there. But just to characterize it very quickly, this is the lakes region of Chile, and it's very fisheries dependent. So I come from the marine world. I'm new to Idaho as of June. And it's been very interesting to see similarities across these different systems that are so different, yet similar in some ways. So there's many fishers in this region, 24,000 in a very rural region. But what this region's facing is aquaculture. Large-scale aquaculture is moving in, and the climate of the ocean is also changing. So sea surface temperature is rising, and they're actually facing really large-scale harmful algal blooms or red tides. So why this relates to Idaho is when we think about mapping environmental change and then mapping values, we have to understand and look at data of environmental change long term. So one way to do that is through what Donna was doing, that Shawn talked about with drones and getting stream data. I've used Landsat data from NASA, and to really understand what is the warming trend. So in 2016, they had an enormous red tide. I don't know if anyone heard about it in the news, but it killed every marine animal possible. But so I was interested in what is triggering this and what are the anomalies. So I used data going back 20 years, and all of this red is an anomaly, which means that is different, very, very different compared to the 20-year mean. So as the more red it gets here, the hotter the sea surface temperature is, and the hotter it is compared to the long-term mean. And then this is chlorophyll data here. So all of those really red areas is really high chlorophyll concentrations, which trigger harmful algal blooms. So this informed my understanding of what's actually happening in the region, which we're trying to do with GEM3 through looking at environmental change long-term. And then you just keep seeing that it gets hotter and hotter. So we're seeing long-term trends here. So there's an environmental change happening, and then people are deciding about how to respond to environmental change. We're experiencing that in Idaho as well. And one of the ways that we can understand environmental change is, one, get out and talk to stakeholders, gather qualitative data, which is this social data that is not quantitative. Well, you can eventually, but put numbers to it necessarily. But that helps us understand the context in which we need to adapt and these conflicts that arise. So this conflict that arose between the indigenous community and the fishing unions was very, very contentious, and I went to the literature, and values often drive conflict and adaptation and to environmental change. And so I ended up doing a survey and looking at values specifically, and I found that two values in particular were very divergent. So divergent, in fact, that when you look at the aggregate of these values, they look like all their values are different. That their value systems are different. And these values were power over resources, so who should have power over resources, who's in charge, and then who has control of the environment? Do humans have control of the environment? And the indigenous community said humans do not have control over the environment. And fishers who are non-indigenous said humans overall, they kind of were split actually, but do have control over the environment. Maybe that doesn't sound surprising. But one thing that they did share was very strongly a shared tradition. So they need access to marine resources in order to continue their traditions for their family and their religious reasons. So, as I said, when you aggregate them, you actually see very clearly this divergence. So this is the non-indigenous fishers and fishing unions, and this is the non-indigenous fishers that are not in fishing unions. They are together in their aggregate of values when you actually do some multidimensional scaling. And then this is the indigenous community members, and they are separate. So this white space is showing how their values are actually very divergent, or what we would think of their value systems. And that's really important because when we think about designing adaptation plans and management for any type of resource, if you have a community of 3,000 people that are really disagreeing about how you should move forward, that's going to affect conservation initiatives, and it's going to affect long-term adaptation. There are ways that you can move through these problems. I haven't done it yet, but one way is to holding things like stakeholder advisory groups or focus groups where you can actually sit people down and have conversations and try to create a kind of a central vision. But what's really important is that these different value orientations, and that's what Haley is going to look at here in Idaho, are there different value orientations for sagebrush, and how does that affect conservation? These different value orientations really come out in this qualitative data. So we can see it in the quantitative data. Oops. Here. that they're divergent. But then we're going to ask, why are they divergent? Well, when we go back to qualitative data, we see that indigenous individuals in the indigenous community are very much conservation focused, and they see the environment as a reciprocal relationship that they give and they take, and the environment gives and takes. Whereas fishers that I work with that are non-indigenous are very resource focused. So they think if they change management controls, they have more control over the environment. And that's just setting up a situation where the adaptation plan moving forward or preferences for management are they're just struggling to find a way forward. And they're still struggling, but they're still trying to work together. So I'm watching this long term. But where this connects back to Idaho is when Haley's looking at sagebrush and values, and we think about how it's getting so fragmented. If people have divergent values across a landscape and we map those values, we can start to see if potential conflicts arise. And if it does, we have to think about how we can resolve those to move forward for the betterment of sagebrush, for example, but also of human systems as well. So that's it.
All right. So we're gonna ask the speakers, John, Jen, to come up here on stage. They're gonna grab mics, and we've got about 23 minutes for questions from you all. And you can ask to somebody specific or a specific question, or you can ask and I can try to figure out who might be the best person to answer that. And feel free to ask anything related to the presentations, or have comments. That's true too. And we do have, let's see here, Leo and Sarah in the back are going to help us. They have mics so that when you ask your question, we'll be able to hear it clearly. So if you want to just raise your hands if you have a question, say it right up here in front.
How do you know the plants are talking to each other, and what do they say?
Yes. So we haven't done this work, but others, what you can do is you can damage them. So you can pretend to be a sage grouse or an insect, and then you can capture the headspace, the volatiles that get released on little cartridges, and then we bring these back to the lab, and then we measure the chemistry and the different peaks, concentrations, that's their message. And so we can then compare the message before you got browsed or fed on or experienced drought or whatever stressor you want, and after, and the difference between those two chemicals tells us what they're saying. So we're still trying to translate. We actually have started working with engineers to create little tags. These are graphene patches, so we can eavesdrop on sagebrush. So we can put a little probe in and turn on genes, and then we can measure on this graphene patch the message that comes out because it's just an electrical signal. So we're trying to do it in real time to find out when the message is loudest and when it's the quietest, and then how other plants respond. So we're getting there.
Great.
That was my question, is how do other plants respond in terms of if you are being nibbled on by an ungulate, for example, and so you send off your cloud of, oh no, into the air. Do the other plants do something to prevent browsing on themselves? And if so, then why would ungulates survive that? Like how do you survive if plants are sending up icky things?
So your upwind plants can't hear the message. So browsers should move upwind, I guess. We haven't tested that. And the other thing is that so they can hear each other within about a half a meter of one another. So because what happens is the plants actually induce their chemical defenses. So when you hear it, genes turn on, they synthesize new chemicals, and then now the plant is more protected. And there are other plants that can eavesdrop. So wild tobacco actually is not related at all, and it hears it, and it induces its chemical defenses and keeps it defended. So we know this because plants, after they've heard sagebrush, have fewer insects that attack them. And if you bag the plant, so if you damage it and then put a bag around it and silence it, nobody responds. So that's the evidence that we have so far.
That's interesting. Okay.
This is kind of a general question. So it seems I don't want to use it. It seems that you're 2 years into this 5-year program, and it just seems like that's such a short period of time. It's an amazing program. First part of the question is, what happens at the end of the 5 years with all the data and the knowledge and the awareness? So I'd be really curious about that. And the second part is how can we support this endeavor?
Sure. So I'll answer, first of all, the first question. An effort like this, a program like this, isn't intended to just stop, to start and stop. We build in sustainability plans. We look at legacy. How do we sustain the knowledge? Some of it is bringing in people on the program who then apply for more grants and extend and expand and go off in different directions so that it nucleates a bunch of other work. It kind of acts as a seed for further work, for bringing in more students, for expanding our knowledge. So it's kind of using it as a crystal to get things going. So that's the first part. The second question was...
I got a thought on that. There's lots of ways that people can get involved, and we can talk a little bit more about that later. But one of the most important ways is to let legislators know that this kind of science is important, if it is important to you. There's a lot of bad press about science these days, right? About whether it's important, whether it's not important, whether it informs decision making, whether it doesn't. And for people that value what science can do, we need to let people that are decision makers know that. So we actually have very good support on this program from the Idaho legislature, but we would like to maintain that through time, because there will be another EPSCoR grant coming after this one is over, and these efforts will continue, but there will be the next thing, and we need support from the legislature to have that happen.
I'm just going to add one thing because we didn't talk about it, and that's our workforce development plan. So we actually are generating lab modules and year-long courses that are sustainable, that help continue to generate this type of data through our undergraduate and graduate school workforce. So this is both with our predominantly undergraduate institutions and our core research institutions that will have these labs in our institutions, so that every year we're generating this type of data. And then we have a sustainable repository where all this data gets stored and is available to the public.
Okay, this gentleman over here.
Dedicated solely to Idaho, or are other states, surviving states, incorporated?
Yeah, it's a network of 27 states and jurisdictions. Western states include Montana, Nevada, Wyoming. There's a bunch of northeastern states, Maine, Vermont, some southern states, U.S. Virgin Islands, Guam, Hawaii, Alaska. They're all states that have...
I spent my youth in the 40s, 50s, and the 60s in the sagebrush and the streams and the rivers in western Nevada, outside of Pyramid Lake and Reno and so forth. And you come up at the end of the day and you'd say, geez, go back across the street and use the neighbor's sprinklers. Get that smell off of me, because I'm the only sagebrush in the street. But it was great.
You must have been activating their communication networks, it sounds like.
We're talking to you.
Oh, right here.
I got a question. Is this on?
Now it's on. So also, I work here. Hi. I want to put in a plug for the microphones. I know it feels awkward to use them, and it's like I feel very important, so the microphone works. But it is how people who are hard of hearing, who use hearing aids, access the assistive technologies in this room. So if you speak into the microphone, you're actually helping everyone hear. So please do use them. I have a question for Sarah. So I was an anthropology major, and I'm a little rusty on graphs, so I'm sorry. Could you explain how you got to, was the first graph a Likert chart, and then you got to 3D modeling? Could you run that down for me?
I'll just be very brief. And we can talk about it after, too.
Yeah.
But, yeah, so in anthropology, there's multiple different methods you can use to get data for social systems, and one of those is through interviews, which is where that qualitative data comes from. And then you can also use structured surveys, which use Likert data, you've probably used one that says like scale, sometimes 1 to 5, agree, strongly disagree. That's a Likert scale. So that first one was a Likert scale of where it was a 5-point Likert scale. And then the modeling, you can actually quantify the Likert scale across all the individuals and the groups. And I did it individuals and institutions, and then you can create the models of how they're lining up with their values.
And then that makes the 3D chart.
Yeah, well, the program R does that, but yeah.
That's pretty cool.
And I'll just add to that, so those same techniques are used across multiple social sciences, behavioral geography, environmental psychology, rural sociology, and in fact, there's really nice parallels, just a slightly different lens. Anthropology looks at things from a cultural lens, geography from a spatial lens, sociology from the individual identity and and so forth.
Okay. Who's next? Okay, we got a gentleman right here.
Just curious. My name's Craig Hill, but I was wondering why you picked sage and trout. I can understand the trout part, but sell me on the sage part.
Okay. So part of the rationale for these is the long history of legacy data on both genotypes or cytotypes, the knowledge from agencies of where certain seeds have been put and where restoration has occurred or not occurred, the tracking through remote sensing of success, so we call this called demographics, so survival, recruitment, reproduction of the plants. And so because of all that legacy data that goes back 20, 30 years sometimes, it created the data that can immediately go into models that then we can say, is what we expected occurring now so that we can make better predictions about the future. So part of it has to do with the enormous amount of resources that have gone into sagebrush and its conservation in Western states. So that's part of it. The other is that because it's so complex, it's a genetically, it's a good model, non-model organism to give us the capacity, because everything else, at least on the plant world, will be easier than this. So if you're the capacity that we're building in terms of taking these genomes, understanding what plants are saying and doing, we can now apply that technology to other plants that people might have a greater value in. So what we're really building, comes back to that sustainability, is for people to say, now I care about bunchgrass, or now I care about this other plant, or I care about this other species. And we say, we know how to do that. We know how to do the genetics, we know how to map it, we know how to model it, we know how to talk to stakeholders. We've created environments. And so that's part of what we're trying to do is leverage this investment that everyone's made to use these as almost turning them into model systems to demonstrate what we're capable of doing.
Yes, sir.
Thank you. An anthropology question, because it's probably difficult. When humans have disparate values and are trying to solve the same problem, that's what you were describing in Chile, and didn't really come at the solution, but you certainly showed how the values were disparate in your work. This is not unique, and there's a new field of environmental psychology looking at how these problems could be solved, but it's still challenging. Storytelling, trying to share information among disparate groups to try to have them understand each other, certainly. But of course, a scientist thinks that you look for root cause and then try to come up with a solution that solves the root cause. But many people don't believe in that way of thinking. So just wondered if you have, since you're all actually going to be facing this in your work with both trout and with sagebrush in the end, when it comes to working with your people who have varied interests, how you're thinking about working that out once you have data that you're ready to use with sage and trout as well as red tides.
I'll start, but that's a great question, and not the first time I've had it, which means I've thought about it, which is good. So for my work, so one thing that for all social scientists, and I think Andy can talk about this too, is it's really important to disseminate your results in a way that's understandable to the people that you're working with, in a way that's helpful. So, for example, those more like the multidimensional scaling, not overly helpful to the people that I work with, because you also have to think about your population. The population I work with is illiterate. So when I do surveys, they're not on computers. They're me talking to them. So when I disseminate results, and I'm going to be doing this soon, because that research was just done at the end of 2018, actually, but is actually holding focus groups, and separately at first, and talking to them about what I found, because they recognize the fact that they have different values. They already know that, but they wanted to see, this was stakeholder driven, they wanted to see what might be the difference. And so sitting down with the separate groups and talking to them about that, and then they on their own have been holding leader meetings. Is that the right word? Yeah. And I plan to sit down with them actually in March to go over that, about how they need to, like, how they want to move forward. But what you often see for these types of management issues is that people that are disagreeing will end up uniting probably in the next couple of years because they're facing a larger threat, which is large-scale aquaculture. And so that will actually, and has been seen actually in northern Chile, to unite these two groups to move forward with a plan. But what's interesting is that when it comes down to it, they just view their relationship with the environment differently, and that's something that is not ethically my responsibility to change, but it's something I can talk about with them. And I think when it comes to groups in Idaho, and I'm new to Idaho, so I don't want to speak out of turn, but it would be like having these stakeholder advisory groups, having these collaborative meetings where we're actually co-designing research questions and co-designing research, even if there might be conflict across individuals or institutions within the group. And I think long-term, so I just very briefly, when we think about management or governance of resources, we often think about it as an outcome. Like we get to this place and it's an outcome. But really we need to start thinking about governance and management as a process that's moving and continually influenced by change, whether it's human or ecological change. And if we look at, as scientists, governance as a process, we can step back and not just see end results of values, but we can see how those values and how those systems change over time and how those might end up with, from a scientific point, in management decisions and how we effectively communicate that to the groups we're working with. So in Idaho, probably Andy can speak to Idaho better.
I mean, one thing that we have quite a bit of experience doing and looking at, including and are developing in this GEM3, is a number of ways to integrate, to bring together knowledge, to bring together data, to bring together perspectives. And two ways we're doing that and can do that. They all have their limitations. One is using mapping. Looking at things on maps in the landscape provides a common reference for many people, and even from widely differing viewpoints. I've previously done work in the North Columbia Mountains of Canada with major land use conflicts between stakeholders, and eventually using sort of the collaborative, talking with people, looking at maps is one kind of story and narrative that you can use. Another approach that we're developing are, you heard the term scenarios. Scenarios being, what happens if in the next 20 years this happens? And that allows us to align trout and sagebrush data and knowledge from the project, landscape mapping, and explore stories for what might happen in the near future. So mapping and scenarios are two kind of what we would call integrating approaches. And as well as the in-depth knowledge that we have as part of this project, there's some phenomenal science being done. We're also cognizant of how they cross over and how we make the ties with that knowledge and expand it and make it connect to stakeholders and to decisions. Sure.
Can I say really briefly one thing?
Yep.
Really briefly, because of your storytelling. So there are scholars that use narratives of different groups, and people have different narratives depending on what cultural group or sociological group they belong to. And these narratives, they can actually analyze to see if there's commonality between them. And then they can use those commonalities within groups to achieve some type of non-conflict. So anyway, I just wanted to say.
Okay, I'm going to put my moderator hat on, and I'm going to ask you all, because don't make me do it. We don't want Shawn to feel lonely on the end here. I'd like a trout question from the audience, or else I'm going to have to get off the stage and ask a question. Somebody give me, there we go, there's a trout question.
I will say that I spared you all from passing out dead trout and having you smell and touch them. So
I do like tasting trout.
I just an update on steelhead and salmon in Idaho today.
So we've had a couple rough years of steelhead returns to the state of Idaho, and some of you that are pretty actively interested in harvesting them or just seeing how they return and thrive in the system are probably tracking this closely. This is probably one of the worst years of steelhead in the last 2, if not 3 decades of steelhead to the system. There's a number of reasons for that explain that, or at least there's a lot of theories that explain that. And much of that has to do with changing ocean conditions. We heard about some of that today from Sarah, and some of the sea surface conditions, and they're not conducive. And here, of course, these fish aren't migrating to Chile in the ocean, but there are waters off of the Pacific Ocean here. But then we've also had some really rough drought years in 2015. Some of you may remember some stories of the really warm drought that was occurring throughout the area. We had severe die-offs of steelhead. Sockeye salmon were dying in huge proportions as they were returning to the system. And so now, as we're experiencing these fish that would be, say, 4 or 5 years later after these severe events, we're seeing what happened to those fish. They had very low survival years ago, and now we're seeing very low returns. This isn't the first time, though, that these fish have had, faced these adverse conditions. And so we do anticipate, if we continue to take care of these systems, take care of their resources, and provide them adequate passage, that they'll continue to rebuild. And in some cases, they may need support, more support than others, though.
So we're really pretty much out of time. We've got time for one last short question. Okay, right here.
So do the trout talk to each other?
Good one.
There's definitely a lot of communication between trout and how they sense each other. They have a interesting feature called their lateral line. If you've ever held a fish, they have a line on the side of their fish, and that actually they use that for schooling behavior. And when they move, they can sense movement in the water using that lateral line, not only between the individuals, but if there's an organism entering the water, if there's a change in flow conditions. And then, of course, there's in their spawning stages when they're actually mates or males in many cases fighting for access to females. That's a different form of communication, of course, but there's many forms of communication that animals partake in that are quite different than plants, certainly.
Okay. Thank you. Thank you all for being here and coming to talk to us. And seriously, thank you all, because this is also a way that you can support this science, right, is by being here tonight. We really appreciate you all being here. We wouldn't be here without you. And also, this was Nicole's idea. I'm going to put her on the spot. This was not my idea. This was Nicole's idea. She was like, well, if you're going to come here to do this other thing, why don't you bring a bunch of scientists to talk about what you guys do? This is a community that will appreciate that. So thank you to the library and to Nicole for allowing us to do this and making this possible.